I want to welcome everyone to the 2023 QBI Internship Symposium. It's great to see this, robust set of speakers. We were doing this before the pandemic, and then it kind of died down, and it's great to see it come back in full force here this year. So we had 14 different young scientists in this program over the summer, and we're lucky to get 11 out of the 14 presenting here this afternoon. And each presenter is affiliated with a program or an initiative. So different programs and initiatives that are being represented here today: the HARC Center, focused on HIV, the Cancer Cell Map Initiative, the QBI YES program, QBI DARPA, and the AVID antiviral discovery program, and PUMAS are all being represented here today. So I think this is a great representation of the future of science. We've got a great group of speakers, and I will pass it off to Robin, who's the first chair.
Okay, hi guys. I am going to be our first session chair, and I'm very excited to introduce our first speaker, who is, we have a hybrid symposium today. So the first speaker is going to be on Zoom. This will be Simran Paralekar, and they were sponsored by HARC and mentored by Ujjwal Rathore. And today, going to present with a presentation titled Automating HIV Gene Connections and Network Propagation. There it is. Okay, great. And do you have a camera so we can, I don't know, Gina, is there?
Hi.
No? Okay, maybe no camera for this, but that's okay. All right, go ahead, get started. Thank you.
Okay. Can everyone hear me?
Hi, yes.
Okay. Hello, everyone. Okay. Hello, everyone. My name is Simran, and today I'm going to present to you all about what I did this summer: automating HIV gene connections and network propagation. Before I get into the project, I'll introduce myself. I'm a high school student from Seattle, Washington. I'm interested in computational biology, and some of my hobbies are playing the flute, the ukulele, baking, and singing. First, I'd like to give a little background on HIV. It stands for Human Immunodeficiency Virus, and it is the causative agent for AIDS. This is a virus that attacks cells that help your body fight infections. The first reported case of HIV was in 1981, and there's no current cure for HIV, but there are various medicines and treatments that can control it. As of 2022, there are 39 million people living with HIV. This speaks to the importance of discovering more about HIV and working towards finding a cure. The team that I worked with in the Marson lab have been doing exactly that. They're trying to understand more about the mechanisms and pathways of HIV infection. When I came to the lab, the team I was working with, including my mentor, Ujjwal, had already developed a method for CRISPR gene editing in actual human T cells, which wasn't done till recently. They isolated and activated human CD4 plus T cells, then infected them with lentivirus containing Cas9 and other lentivirus with guide RNA. The team both overexpressed and knocked out all 20,000 human genes. They then infected those edited T cells with HIV. There was a GFP tag on the HIV, so more HIV infection meant you would see a certain gene to be more in like the GFP positive region. Using this method, Ujjwal and his team were able to identify top HIV host factors. As you can see on the bottom graph, it says restriction factors and dependency factors. Restriction factors are factors that restrict HIV infection, and dependency factors are factors that HIV depends on to infect a cell. The graph below shows how the cells with certain gene deletions and overexpressions were sorted by GFP. Now that the top gene hits were retrieved, literature reviews needed to be made by taking each gene and manually searching up that gene's connection with HIV. The problem was that these were large numbers of genes either increasing or decreasing HIV. This technique would have taken weeks to complete. It would also take longer to find the most interesting genes. Automating this process would reduce the time needed to find HIV gene connections. To solve this problem, I wrote code that could automate the process of finding HIV gene connections. Above is an example of the output from my code, the CRISPR-A restriction factor table specifically. The code reads the keyword and retrieves the PubMed IDs of articles that mention that keyword and HIV. Also, PubMed is a research article database. Then the code reads every other alternate gene name and does the same, adding more PubMed IDs to the last column. For example, I take MUC1 and HIV and try to search for articles that mention both MUC1 and HIV, retrieve those PubMed IDs in the last column, as you can see, but then it also take the alternate gene names such as PUM and HIV and do the exact same thing. If no IDs are found, either not found or not known is written. I had to modify the code quite a few times. There were a lot of PubMed IDs for some of the genes, so I had to write code that restricted them to just search the abstract and ignore empty abstracts to increase chances of true positives. I've created literature review tables for CRISPR-A and CRISPR knockout restriction and dependency factors for the top 50 hits in each of the 4 categories. This code can work with any list of genes, alternate gene names, and another constant keyword such as HIV or a protein or another gene. It is also easy to switch out the PubMed database with another. You would need to do research, though, on various packages that would work for this new database. To mark the most interesting genes, I've been using the generated tables and string database, which maps gene interactions. This is an example of finding the most interesting genes using string database and the generated tables from my code. The first 3 bullet points are from one row in the CRISPR-A restriction factor generated table. As we can see, MUC21 has no known connection to HIV on PubMed, meaning it hasn't been studied in the context of HIV, but on string, it shows that it does have a connection to MUC1. On the generated tables, MUC1 has a known connection to HIV that has been studied in the context of HIV. After additional research, I found that the MUCs are an important part of inflammatory and innate immune response, but the expression of these by viral infections is still largely unknown, making this an interesting gene for further research. The other thing I did this summer was work on a network propagation project, which I'm still working on visualizing. Max Bennett, an undergraduate intern working in the Krogan lab, shared a shortest pathway analysis network code, which he made. It takes CRISPR gene hits and generates a map of the shortest paths between those genes and HIV host PPIs, or protein-protein interactions. This code uses string database to expand the PPI and CRISPR gene hits network. I used this code to find the network between our list of top hit genes and 435 human genes, which had connections to viral HIV proteins, as shown by the orange circles. But I'm still working on visualizing that network. In the future, I could automate the process of finding interesting genes, like I mentioned before, using string database, so we wouldn't need to analyze this information manually. Also, finding gene connections with specific HIV proteins could retrieve a more focused list of PubMed IDs. Finally, in the future, as I mentioned, I could continue trying to visualize that network that I was working on in the last week of my internship. Thank you for listening to my presentation. I'd also like to thank Ujjwal and Eli, my mentors, who taught me a lot about the research and how to apply computer science to biology. I started learning the coding language R this summer, so I was excited to have been given the opportunity to learn from so many knowledgeable people and work on impactful projects. I would also like to thank Ishan and Nandini for teaching me lab techniques. It was my first time working in a lab, and I'm grateful to have been given the opportunity to do some wet lab work under the mentorship of these interns as well. Thank you to Max Bennett for teaching and working with me on span analysis. I'd also like to thank others like Ben, Alex, Marcin, Emin Krogan, and Jyoti as well. And of course, all the interns for adding to the social aspect of work, and everyone in the Marcin and Krogan lab. Lastly, I would like to thank HARC for sponsoring me. Thank you.
Okay, thank you for a really great talk. Are there any questions from the audience? Maybe I can start. So that's really cool. I love how you've automated basically literature review, which I think most of us don't really enjoy doing all the time. In addition to pulling out the PubMed IDs, could you tweak your code to pull out maybe even titles or like full abstracts, or do you think it's specific to numbers?
It doesn't search the IDs. It searches like an entire article. So exactly like you said, before it was just searching the entire article page, but now it searches for those specific keywords, like the example before, MUC1 and HIV in the abstracts only of those articles.
Yeah, and could you have it report back those abstracts of the ones that were matches?
Yeah, that's
Yeah.
through, yeah.
Very cool. Yeah, Einar has a question for you. Maybe you wait for the microphone.
Hi, that's a really cool project. Thank you. I was wondering, would you be more interested in looking at the hits that are widely described in literature, or rather more interested in the ones that are not that widely described?
Probably the ones that aren't as researched yet. That's sort of what I was doing after I generated those tables, by finding indirect connections. For example, like the MUC21 and MUC1. MUC21 had no research done on it in the context of HIV, but there was an indirect connection through MUC1. So ones that haven't been really studied yet.
That makes sense. Thanks.
Yeah. We have one more question from Manon.
Hi, great talk.
Thank you.
I was wondering if you looked at the distribution of how many hits you got for the different, how many PubMed ID hits you got for the different CRISPR-A hits, and what that looked like what the max was, like how many was the most papers that came out, and how did it, was it distributed?
Initially, it was a lot. It was maybe like 100 for each gene, and some had none or very little. But over time, modifying the code by just searching the abstracts or excluding empty abstracts and restricting it to just 20 sometimes to help cut it down just to 20, just a range from either 0 to 20 PubMed IDs.
Great, so that it's actually manageable for a human to then look at it afterwards.
Yeah.
Perfect, thank you.
Any other questions? Okay, let's all thank Simran one last time.
Thank you.
Thank you. And we will, I'm happy to introduce our second speaker, who will also be virtual. Our second speaker is Angelo Evans, who's also sponsored by HARC. They were mentored by Catherine Jackson Jones, and we'll be talking about understanding the interaction between HUE1 and VIF. And without further ado, and they're in the Hultquist lab this summer. And without further ado, let's all welcome Angelo.
Hi, thank you. Everybody can hear me, right?
Yes, we can hear you. Yeah.
Perfect. So my project this summer was understanding the interaction between the human protein HUE1 and the HIV protein VIF. A little bit about myself before we get into the project. I am a second-year undergrad student at Loyola University, pursuing a molecular biology degree. I have had previous lab experience in this lab. This is my third summer so far. And my project is just focused on the protein interaction between HIV and host proteins. So we first got to this project by looking at two other experiments funded by HARC. The first being my mentor's experiment that looked at the knockout of HUE1 in primary CD4 T cells and seeing how that affected the infectivity of HIV. So the first two see if I can point to it. There we go. The first two figures on your left are 2-day post-infection and 5-day post-infection. When we look at the HUE1 knockouts, we can tell that infectivity skyrockets in both of these settings. And then we also looked at, to more narrow it down on how HIV or how HUE1 is interacting with HIV, we looked at another former figure paper, which told us that HUE1 is actually interacting with VIF at a pretty high rate. The questions we gathered from this were, is this interaction between HUE1 and VIF the reason for the increase in infectivity? And to figure that out, we are looking at what rate and what importance each HUE1 domain is interacting with VIF, as well as does that, do those different domains have different restrictions on the production and function of VIF? So to start this project, I cloned a series of deletions of HUE1, 4 from the N terminal going down, and then 4 from the C terminal. This is just taking out certain domains of HUE1. So all of the N terminal constructs have these 2 first armadillo regions, which are known more or less for interacting with other proteins. And then the C terminal constructs have all of the HEC domain, which is usually adding ubiquitin onto other proteins to signal to the cell. After those constructs were completed, we co-transfected HUE1 and VIF into HEC293 T cells. VIF has strep bound to it, and HUE has flag bound onto it. So after we gathered those and lysated the proteins, we then ran a affinity purification using streptavidin beads. This way, if we're pulling down on VIF and we're still getting HUE at any rate, we know that the regions or the domains in that construct are important to the interaction. Then we ran all of these on a Western blot to check expression and if the 2 are actually having an interaction. The first set was the N-terminal constructs. From left to right, you're seeing the input, the IP or the affinity purification, and the flow through of those experiments. So starting with this first probe of our flag, this is to check if we're having any interaction. So if we're probing for flag on these Western blots and we see any of our HUE constructs, any of these, we know that they were pulled down successfully in the IP and they have interacted with VIF. So we see bands at every level are full length HUE1, which is just a positive control, since we know that those are supposed to interact with VIF. And then each one of our constructs, with the 4th construct being the lowest expression rate. And then the last column is always just a negative control, making sure that there's no non-specific interaction between VIF and other proteins that may have flag on them. In this second row, we reprobed for strep. So looking just at the VIF levels of expression, and we found that in our environments that had full-length HUE1 and VIF, and our 4th construction in VIF, this is this one that has a tiny bit of the HEC domain, we found that we are getting an increased amount of VIF expression, as well as possible monoubiquitination of VIF. So it could be HUE1 might be adding a ubiquitin onto VIF, but we're not sure of that yet. We also then probed for CBF beta because going back to one of our previous papers, we saw that CBF beta is recruited by VIF in host cells for stabilization, as well as to form another construct to help VIF in its actual function. Going on to the second set of experiments, the C-terminal constructs, we probed for flag again and saw a very similar outcome of every HUE1 construct seemed to interact with our VIF proteins, and then reprobing again for strep to look at the VIF expression levels. We don't see that much of a change in expression, but we do still see in the HUE1 full length in the VIF environment that we are still getting that possible monoubiquitination, which is telling us and reconfirming that that wasn't just a fluke in that first set of experiments. Again, we reprobed for CBF beta to make sure that we're that there's still that interaction between VIF and a protein that we know it should interact with. So from that, we can tell that the existence of the armadillo regions and the HEC domain don't really matter in terms of interaction between HUE1 and VIF. HUE1 can include one or the other and still bind to VIF. But it does affect this possible ubiquitination that we see in some of the environments. Because only when all three of those domains exist on HUE1 do we see that ubiquitination. So next steps, we first want to look at our interactions between HUE1 VIF and CBF beta. To do this, we are going to rerun these experiments, and in the affinity purification step, use flag tag beads instead of streptavidin. This way, we're pulling down on HUE1, and we know that in this set of experiments that HUE1 and VIF, no matter what constructs we used, interacted, so we should still pull down VIF, but we want to get a more clear picture of if these three proteins, HUE1, VIF, and CBF beta, are in a construct together, or if they are in some way competing for interacting with VIF. We also want to create another series of HUE1 constructs. The constructs that we made this time were more focused around the deletions of our two armadillo regions and our HEC domain. Now we want to focus more on what if those are both completely gone, and specifically our UBA, WWE, and our BH3 domains within HUE1. We lastly, at least for these constructs, want to transfect them into more HEC cells, but also with HIV plasmid to see if these constructs have any restrictions on HIV production. I'd like to thank first Judd Holquist, that's the PI of this lab. He's allowed me to be present in the lab and do wet lab work for the past 3 summers now. And then obviously Catherine has been an amazing mentor through this summer. And then obviously the Park Center for funding this project and making this possible.
Okay, thank you for a great talk. Does anyone have any questions? Maybe we have time for 1 or 2 quick ones. Yeah, Einar has a question.
Hi, good talk. I was just wondering if I got this right at the beginning. Huey1 was a hit in the HEC293 screen and not in the JERCAT screen. Is that right?
You're referring back to this slide, correct?
Yeah, exactly.
Yes.
Do you have any
It
thoughts
was
about?
only in, sorry.
No, yeah, exactly. It's only in HEC, right?
Right.
Yeah. Do you have any thoughts about why that is?
We didn't focus too much on that aspect of this. I am not really sure why that would be. I've only worked with hex cells, and so I'm only familiar with that that environment of growing cells and proteins. So
Yeah. Okay. No, I see. It's a tricky question and a tricky thing to think about. So I was just curious. Thank you.
We have one more question.
Thanks for a cool talk. So I know about Huey1. I think it's an interesting protein for some of the work that we do in the lab. And I'm just curious, do you know if the VIF interaction alters what Huey1 does, or is it really just VIF taking the ubiquitin to go and do its job?
So we don't know the functional side of this interaction. We actually looked at, and I did cut this out of my talk, we looked at what the environment between a catalytic mutant Huey1 that we just stopped the function of the HEC domain and that interaction with VIF would look like. And I can pull that. And it brings up a lot more questions and makes it a lot more unclear what the functionality of this interaction looks like. There's maybe 5 questions that came from this one experiment.
Sounds typical. Thanks very much.
Yep.
Okay, guys, in the interest of time, I'm going to push on to our last speaker for this section. I think yeah. So please join me in welcoming Julie Bilodeau. She was mentored by the Gordon Lab, John Gordon. Yeah, I can help you set up your computer as well. Should just be. Oh no. Is it not there? For those of you who can't see, she will be talking about overcoming toxicity to target oncogenic protein kinase A. And hopefully, Gina, do I need to press anything? Oh, perfect. Okay, great. Okay, please join me in welcoming Jolie. Hello, everybody. Today I'm going to be presenting my presentation called Overcoming Toxicity to Target Oncogenic Protein Kinase A, which I will be referring to as PKA in this presentation. So first off, a little bit about me. My name is Julie Bilodeau, and I am a high school senior, and I'm an intern at the Gordon Lab. I don't really have any previous biology experience, so this time in the lab was meant for me to kind of try and figure out, like, you know, what's going on there. For some background information, the Gordon Lab focuses a lot on fibrolamellar carcinoma, which is a liver cancer that is found in those between the ages of 12 and 40. It does not respond to common cancer treatments, and so what most of this presentation is going to be is kind of trying to figure out a way to cure this. So there are 3 main concepts that are going to be important in this presentation. The first one is isogenic cells. These are cell lines that are manufactured to be completely identical except for one thing, and that's the thing that we're going to study.
The second concept is a therapeutic index. So up here, you can see an example, but basically that is the difference between a drug's effect on cancer tissue and on regular tissue. And then the third concept is bivalent drugs. These are drugs that are linked, so two different compounds are linked, as you can see here. And this is used to increase the specificity of what you're targeting. So the following experiments will be performed on FLX1 cells. This is a cell line that was taken from someone with fibrolamellar carcinoma. And this cell line has actually been modified so that when doxycycline is added to it, PK regulators will basically come and start regulating again, because with this mutation that you can see down here, so this occurs in people with fibrolamellar carcinoma, and it basically stops PK regulation, meaning that cell growth is basically uncontrolled. So when there is no doxycycline, they act as normal and are uncontrolled, and when there is doxycycline, they become regulated once again. So PK is basically, it affects cell growth, cell cycles, and cell migration. So what we're going to do is try and use PK inhibitors, because we can't just completely get rid of PK because it's so important. What we're going to do is use PK inhibitors in order to destroy as much of the cancer as possible without negatively affecting or killing the host. So the first experiment that I did is we took 4 96 well plates, filled the outer wells with PBS, and then we filled the first 3 rows, as you can see here, with FLX cells without doxycycline, and then the bottom 3 with FLX1 with doxycycline. And again, this is what it looks like without it. It means that PK is unregulated, and when it is added, PK is regulated again. So these are the structures of the PK inhibitors that we're using. These two look similar here, and this one looks completely different. So these are our results for the first experiment. All 3 of these have some kind of therapeutic index, but the one that is really shown the most is right here. Another way of thinking about it is the area under the curve. So here we can see there is a larger therapeutic index than here or here. So for our purposes, this is the drug that works the most effectively. And then we are going to move on to targeting the EIF4A protein, which is also associated with fibrolamellar carcinoma. So unfortunately, EIF4A is a very, very common protein. And so in order to specify where we're targeting it, we're going to use bivalent drugs. So the bivalent drugs that we're going to use have 2 compounds, racaglamide, which targets the EIF4A protein, and EFT508, which targets this right here, MNK. And basically what we're going to try and do is get racaglamide to target these EIF4A proteins and not these ones, or any other ones. So this is the structure of all 4 drugs. This is racaglamide on its own. This is racaglamide and the other compound. You can see them in all 3 of the other ones. So the difference between these 3 drugs right here is the linker length. So here we have a shorter linker length. This is our minimum. This is an average linker length, and this is our maximum linker length, because anything past that doesn't really work. So what we're trying to see is basically what range of results we will get. So our second experiment is set up exactly like the first one, except instead of adding PK inhibitors, we added 4 different EIF4A inhibitors, or I guess 1 EIF4A inhibitor and 3 bivalent drugs that also target that. And our results were that here we can see that the drug with the longest linker length, SW134, was actually the least potent, while the one with the shortest linker length was the most potent, even more so than ROC itself. So what we can determine from that is that the more linkers there are, the less potent the drug will be, which is what we expected to find. So this is a comparison of here ricaglamide with and without dox, and then here SW134 with and without dox. So here we can see that ricaglamide doesn't really have a difference. There's not really any kind of therapeutic index between these two. But for SW134, we can see that there is definitely a therapeutic index when you add doxycycline and when you don't add doxycycline. And so that tells us that there's definitely potential with these bivalent drugs because they are working to a certain extent in terms of more specific targeting, which is our overall goal. So the conclusion is going to be that bivalent EIF4A inhibitors do have potential because they've shown us that they can have, even if it's small, a therapeutic index between mutated and normal cells. And that means we can start doing more experiments with perhaps more expensive equipment and stuff. So our future direction is also going to be characterizing the drug's effects, so exactly what they do. And we actually have already done this. So we did a Western blot and the results that came out were exactly what we expected, which tells us that the bivalent compounds are actually working. And that's a good thing. So for my personal conclusion, I really enjoyed working at this lab. I met a lot of different people, and I think I've really learned a lot about like the scientific community, and it honestly has made me more interested in perhaps doing that later in my life. So I really appreciate that. My acknowledgements, thank you so much to my mentor, John Gordon, and Brian Pasquale, and all of the Gordon Lab who are all over there. And thank you to the Showcat Lab for providing us with the bivalent compounds. And thank you for everybody for listening to my presentation.
Okay, great talk. Does anybody have questions for Jolie? Okay.
That's really cool project and good work. I was just wondering, you showed that you have a little bit of a therapeutic window with the 34 linker length compound. Did you also check the ones with a shorter linker length?
Yeah, so we did, and it's just like there wasn't really any. I mean, it was just so incredibly small. This is just the one that worked the best, like in terms of making a therapeutic index.
Do you think it might be reasonable to increase the linker length even more to make this therapeutic index larger?
I think if we increase the linker length, the drug might, let me know if I'm wrong, but the drug won't be able to enter the cell as well. So it just doesn't make it as, yeah, it's just not as effective.
That makes sense. Thanks.
Any more questions?
Hi, really nice talk. I have a very simple question because this is very far from what I do. But why do you have the PBS around the edges of the 96 well plate?
That's a great question. I actually do not know the answer to that.
Evaporation. Yeah. Okay. I think Nevin has one last thing to say, but before he says anything, can we all give a round of applause for really great talks? And that was the end of our HARC and CCMI symposium. And then, so Nevin will give some talk, say something, and then we're gonna have a quick break.
Those are great talks, right? Fantastic. I just forgot to say something very important. I wanted to thank not only the mentors for working with the students over the last few months, but then the team that put this event on, Robin, Shivali, Manon, Kelsey, and then Jean and Alexa in the back, and Phoebe as well. So thanks guys for putting this event on.
Okay, guys, we went a little bit over, but try your best to be back in by 1:50. I know that's only 5 minutes, so really quick break.
Afternoon, everyone. It is my pleasure to introduce the QBI Yes Fellowship Trainees. QBI Yes stands for Youth Equity in Science. The program was designed for students from underrepresented communities and first-generation students. It's an honor to introduce Philippe Morfin. Philippe is a computer science student at Boot College, and he's interested in applying his learnings towards the biosciences. Philippe?
Thank you for the introduction. Like I said, my name is Felipe Morfin Jr. Is it not popping? Let's see. Let's try it again. Is it not working? It was working earlier. Share?
Share. So it's share in call. There you go.
It was working earlier, just to let everybody know. Yes. That's right. Can it not just, can it not just play to, no? Share?
Share in call. Click layout. Click layout.
Because it was working earlier. Like this one? Yeah, if it works for everybody else, yeah. All right, perfect. Well, my project was on the study of structural connectivity for tinnitus classification. I was part of the Department of Radiology. My mentors were Professor Sri Katan Nagarajan and Dr. Sanjay Ghosh. So from where I'm from and where I study, it's a very small town. I don't know if everybody knows, but it's a town named Corning, but they're known for their olives, which is kind of off-putting name. It's 2 hours north of Sacramento. And then I study at a nice college called Butte College. I've just completed my first year of computer science, and I'm part of the MESA program, which is Mathematics, Engineering, Science Achievement, which is, I found this internship. And you're probably wondering, what is tinnitus? You know, you think it's something with the ear, you know, ear hearing damage, something like that. It's actually quite more complex. It's an auditory phantom disorder characterized by the perception of internally generated elemental sounds, perceived as a buzzing, a ringing, anything that's being generated not externally but internally with your brain, it could significantly reduce the quality of life of individuals. It could be accompanied by depression, insomnia, and anxiety. It's basically with this debilitating disorder, it could just add on to the to your mental health. It could just completely be something that's so hard to cope with and so hard to be able to live with that it just starts to add on. And just like how people kind of associate tinnitus as a hearing damage, actually 25% of people with tinnitus have normal hearing thresholds. Now, the data that we've collected was from DTIs, or diffusion tensor imaging, for white matter, and structural MRI for gray matter volumes. As you can see, with the raw imaging that we have taken from the MRIs, we've made it into a matrix to classify the control tinnitus and then keep note of the significant differences between the two groups to see what exactly is happening inside the brain due to tinnitus. Now, you're wondering, how does a computer scientist kind of fit into this bioscience field? Well, I'm actually trying to see how we could implement machine learning for tinnitus. It could advance the previous investigations of neuroanatomical changes associated with tinnitus and provide some insight on how structural connectivity is affected with tinnitus. With the SMRI, we could create an algorithm with computers in order to be able to distinguish the two groups of healthy control patients and tinnitus patients. Now, as you can see, you're probably wondering, well, what's the difference? Exactly that. They're kind of similar. A tinnitus patient and a normal healthy control patient are the same person. The only thing is, there's significant but subtle changes as this part and this part. There's subtle changes that we overlook majority of the time, but can be caught on with machine learning. So this is a great example to prove how machine learning can catch on to these subtle differences without us having to look at an imaging at an image for an extraneous amount of time trying to find out what were the differences. The next step with this project is to be able to train a machine to be able to distinguish the two control patients and be able to have them characterized on a range from severity. Also, another key point about the project is be able to see how tinnitus affects the brain over time and see what can we predict and what can we make out of it and how can we diagnose these patients. My acknowledgements for, well, as a thank you is Mesa for helping me get connected with Gina, QBI for giving me this amazing opportunity, and obviously my mentor, Sanjay. He's been a great help, kind of catching me up to speed to the project and be able to go along with the ride.
That was a great talk. Thank you.
Thank you.
So I don't know a lot about tinnitus, but from what I remember, it can be caused by a bunch of different things, like even infection. Have you guys looked at maybe classifying samples also by like what was the contributing factor for tinnitus?
We were trying to kind of work on the classification part for like the two different from people who we know that have tinnitus, from the people that we know that don't have tinnitus. What we're trying to expand on that classification is also what were the causes for tinnitus? Like, was it something just that happened, just neurological? Was there an infection? Was there something? So that's what we're trying to expand as well and add that to our machine learning to be able to characterize these by different causes of tinnitus.
Very cool. Thank you.
It's cool to see something so close to the clinic. I was wondering, are you also thinking about applying this to other diseases or other conditions than just tinnitus?
Yes, I'm glad that you asked. We recently have been doing it with schizophrenia and Alzheimer's disease. It's just that we wanted to branch off with tinnitus because previously there hasn't been any research done on it. So it's kind of a new ground. We've been doing continuous projects with other diseases like that, like as you were asking. And so far it's been very successful and we're trying to expand on it. And one of them is tinnitus. Thank you.
also. Are you interested in participating in a hackathon? The QBI hackathon is taking place in November.
I'm very interested into it, especially being exposed to biosciences and seeing how I could provide help as a computer scientist. It's been something I'm very interested in. Thank you.
Okay, and our next speaker, we have Belen Magina Velos, and she is an intern with the Fujimori Lab and mentored by Jonathan Sandova. And she is a student at Cuesta College in San Luis Obispo, and saw Manon's and Kelsey's panel discussion and was inspired to apply for this program.
I know. Yeah. Okay, well, hello everyone. As Gina said, my name is Belen Maguiña, and I would like to tell you a little bit more about my summer internship. So, I'm sorry. Well, a little bit more about me. I'm a sophomore at Cuesta College, which is a community college in the central coast of California, where I'm majoring in biology. So I became interested in science since I was a child because of some medical conditions that some of my close family members were going through. So I became more interested about these questions about how why it originates, how it develops, what its evolutionary process is in every human being, but also and obviously how it's solved. So for me, during this internship, I get to know that this part of science, biology, goes beyond the solution, but rather finding the origin to better understand the illness. So this summer, I had the opportunity to enter under the YES program in Dr. Fujimori's lab, where Dr. Sandoval was my mentor. During these days, I had the opportunity to learn more about the career as a scientist, but also to put in practice all the knowledge that I know since college. So Dr. Sandoval is currently working on a fusion protein of nucleoporin 98, which is also known as NUP98, with a C-terminal portion of KDM5A, H3K4ME3 demethylase. So I want to tell you a little bit more about this fusion protein. So the NUP98 KDM5A is a fusion gene produced by a chromosomal rearrangement that also codes for a protein that is part of the nuclear pore complex. So this fusion gene has been related with acute megakardioblastic leukemia, which is also known as MAKL, which is a subtype of acute myeloid leukemia, AML, that usually affects young children, mostly like 40% of the children. So during this internship, I had the chance to help with the expression and induction of 4 different regions located in this protein. So I was also able to learn more about the PCR cloning and protein purification techniques. So these are some videos and photos about my experience here. So the first thing that I did was the growth of bacterial cells. So I did a 1 liter culture of each region of NUP98. So for this, I used at least 10 milliliters of sterilized Illyria broth mixed with 100 microliters of kanamycin, which is an antibiotic, and the corresponding cells. And then it left overnight. So at 37 degrees, so it can grow. Next day, I added each of the growth to each liter of liver broth, which was already sterilized. And I added also 0.05 grams of kanamycin, but this time in solid. And I left it again in 37 degrees until they reach the required optical density. So for 3 of them, the optical density was for 0.8, while the other one was 0.6. So each of them having reached their optical density, I induced their protection expression with chemicals, the IPTG, which is the one who accelerates the growth of the cells, and zinc chloride, which controls the protein. I don't know if I can put the video so you can see my experience here. Well, after that, one of them was left overnight, and the next day the cells of that 1 liter were harvested, while the other ones have to be harvested after 4 hours. So for doing that, I put the liters into other containers so they could be centrifuged. And at the end of this procedure, at the bottom, I can notice the precipitate of the cells, which becomes solid. And so I take the cells from the containers and I put into other bags so they can be frozen for later use. So in conclusion, during this experiment, I only did 1 liter per each region, so we can observe how is the reaction of each region of the protein. Because since this protein is not to study, we can't know how it's going to react or what's the time of expression or protein expression. I'm sorry, and also how it's going to develop this region. So this could be studied further. But personally, I can say that this time here during my internship, it allowed me to have an approach and learn more about this field of work in science. Thank you.
Nice presentation. Thank you very much.
Thank you.
I wanted to ask if you could talk a little bit about what you know of what is going to be done with the protein once it is expressed and purified.
Yeah, well, some of the data that we had from these cultures is that we can determine what is the time for each expression of the region, so we can further know how it's going to work if we replace one part of this region with another one, so we can further know how it works, this protein.
Actually, Manon had like exactly my question too about what you were going to do with the proteins once you purified. I guess I can ask, what did you find to be the most difficult part of this process?
Maybe it's not related to the process itself, but I don't know if you once happens to be through this, but at college, you get to know math at some way, and you use it in exams or in homework in the same way that the professor teach you. So I think something that at the beginning, like, not shocked me, but helps me to understand more about this field is that math can be involved in any way. So for example, to add in these chemicals or weighting the liters or the liver broth or another compounds that I need, I really need to use a lot of formulas that, although I know how to use it in class, I really didn't know how to do it in the field. So I think that's maybe the difficult part.
Yeah, nice talk, and a really nice description of the process you were using for the experiments. it seemed like there are really a lot of steps to go through. So what strategies did you use to kind of make sure you did the right thing at the right time and kind of kept track of everything as you were doing all those complex steps?
Well, one strategy that my mentor taught me was to write everything I've done into a notebook or my cell phone, something I kept the track of everything I did. So if, for example, I get wrong with some part, I know what I did and how to do it better the next time.
Thank you.
Okay, up next, we have Jane Ton. Jane is in the Castor Lab, or interning for the Castor Lab as in the Department of Psychiatry. And she's from Vietnam. So it is an honor to have you on the program. And yes, I'll leave it to you.
Thank you so much for the introduction. Hello, everyone. Thank you so much for taking your time and be here with us today. Thank you, the organizer, to make this possible for us. Today, I will talk about pre-pol inhibition of the acute startle response, PPI. Just a little bit about myself. I was born and raised in Vietnam. And recently, about a few years ago, I immigrated to the United States alongside my family and make my new home in Los Angeles, where I went to community college and got my associate degree in math, science, technology, and engineering, and an associate degree in social and behavioral sciences. And then I transferred to UCLA and graduated amid of the pandemic. And recently I moved to the Bay Area for a biotechnology program. That's where I learned about QBI Fellowship. The passion that I have for science really stemmed from my curiosity about the brain and the idea that I could take part in studying and restoring the central core of what makes human who they are and how they interact with the world is truly humbling and incredibly inspiring. And that's really motivate me to pursue science and medicine. And the opportunity to be part of Castor Lab, understanding the potential deficits of the neural mechanism underlying the neuropsychiatric diseases really align seamlessly with my career aspirations to become a physician. And so at Castor Lab, we study the behavioral variability and how it changes in the context of disease processes, and that towards understanding how the changes in the brain leads to the changes in the behavior. We really try to examine the behavior in its full context, a full complexity and richness to provide an interpretive framework for understanding the neural activities. And so why do we care about animal behavior, right? Because it provides the biomarkers to help track the disease progresses and also help evaluate the effectiveness of the treatment. It also provides the fundamental basis for validating the applicability of the animal models of disease and for interpreting the intricacy of the neural activities. And so at Castor Lab, we study or examine a variety of animal behaviors. The one that I have been focusing on is pre-pulse inhibition of the acute startle response, which I will refer to as PPI. And I will tell you more about PPI in a little bit. But first, let me explain why PPI is such an informative and valuable animal behavior that we should study. PPI has been linked to neuropsychiatric diseases. And an example of that is that people with schizophrenia have shown a decrease in PPI, and even more exciting, with a treatment with antipsychotic treatment, it improved the PPI level. And PPI can also be measured in both experimental animals and human. And so what is PPI, right? Here we're looking as a stimulus in which we always have the background sound at 70 decibel. And then we have a pre-loud sound that will elicit a startle response, right? And with a slightly different stimulus in which there is a softer sound that comes right before that loud sound, it will elicit a startle response that's shown in red, right? So here we are looking at one single startle response of one animal, right? And we measure that across many repeats, across many sessions. Here we're looking at the average movement of the one single animals, right? The average movement of all the repeats. So looking at here, at the same sound level, we can see that the average movement of the one with pre-pulse in red is lower than that of the one that without pre-pulse. And we measure that across all sound levels. And what we see here is that consistently, the movement with pre-pulse that's shown in red is lower than that of the one that's shown in black, which is the one with no pre-pulse, right? And so here we're trying to understand PPI and try to characterize the PPI. And so looking at our data, the sigmoid curve fit our collected data very well. And so how PPI traditionally measure was that they would measure these 2 points only, and then take the ratio, right? And the problem with that is that it's assumed that fundamentally PPI is just the scaling of the y-axis, right? The downscaling of only the y-axis. If we do exactly just that, right, we will get the green curve right here. Now, as you can see, that the green curve doesn't match or doesn't describe our PPI collected data correctly, right? And so what else is there? Well, we can manipulate the y-axis, which we call startle scaling, and the x-axis, which we call sound scaling, or we can combine both of them, startle scaling and sound scaling. And when we do that, we will get the very nice fit curve that fit the PPI data that we collected. And so in the past, our lab has been found the better way and more systematic way of describing PPI, right? Using all these parameters, now we have a more advantage in looking at identifying the differences in different group of animals. And so we asked if this is enough, right? If we have described PPI in its full complexity, and the answer is no. Up until this point, we only measure PPI in adult animals, right? And as soon as we measure the PPI in younger animals, we quickly see that here at the louder sound, there's a greater decrease, right? Greater decrease in younger animal as opposed to plateau in the adults, right? And so this is kind of a surprise to us as new. However, we see it consistently and it's significant in the younger animals. So we see a great decrease of the startle reflex in younger animals as compared to the adult. And so with a more enhanced and added parameter, we are able to capture the drop in the younger animals. And so this is continuing to be the project that I'm working on, to really try to understand what is the implication of this greater decrease in the younger animals, and what it means in the context of neuropsychiatric diseases. In summaries, pre-pol inhibition of the acute startle response can have the potential as a biomarker for neuropsychiatric diseases. It can be better described by both startle scaling and sound scaling, and the greater decrease of startle response in younger animals added the layer of complexity to PPI. So I want to thank you, QPI, yes, for an exceptional opportunity to learn and to explore research. I want to thank my research mentor, Dr. Davis Kastner, for his great support and guidance. His mentorship has been instrumental to my growth professionally and my understanding of the research. Eric Miller has been doing a lot of data analysis. Grace and myself have been collecting a lot of data in addition to data analysis. And of course, I'm always very thankful to the conversation that I have with all the talented members of the lab. Also, I'm very thankful to the various funding sources that make my learning experience possible. And with that, I would love to take any questions.
That was a really great talk. Thank you. I was curious, how do you guys decide how much further in advance to do the pre-pulse sound? Like, have you tried putting it at different times before, or is it like that's just the optimal early time point?
Yeah, absolutely. So currently, we deliver the pre-pulse at 200 milliseconds. So we start the clock, and then 200 millisecond in, we deliver the startle sound. The pre-pulse sound, and then right after, about 100 millisecond delay, we deliver the sound that elicit the startle response.
Are you measuring any activity with the pre-sound as well?
Yes. So let's come back to this slice, right? So, okay. So right here, this is the sound that will elicit the startle response. And this is what it looked like when the animal startled, right? Without pre-pulse.
Yeah.
Now with pre-pulse, which is this one,
I meant like at that exact pre-pulse, nothing happens with the lower sound. It only happens with the startle sound. Or do you not measure that pre-pulse sound?
That 100 millisecond is really short, but
okay, yeah.
I would let my my mentor take that.
I see. Cool. Thank you.
Thank you so much.
Thank you. Great talk. Thank you very much. I have two questions. One is a little cheeky. I don't know. So I couldn't help but notice that there was not just a difference between the young and old rats with the pre-pulse, but also just the general, like for the loudest sound, it just went down, right? Does that explain that I startle much more than my toddler does when he screams at the top of his lungs? I don't know how much 120 decibel is. Would a toddler get there? I don't know. Hopefully not. Okay. Well, so that was the just for laughs, pretty much. And then I was wondering how you actually measure the movement, so how the rat is situated and all that.
Yeah, absolutely. So we have a box. We put the animal in a box, and then there is a sensor at the bottom of the box that will capture the movement of the animal and send to the computer, and that's how we code.
It's fascinating work, thanks. I was wondering, so the PPI looks very different in young animals. Do your results suggest that, or allow you to perhaps measure any neuropsychiatric diseases earlier on, now that you understand that PPI might look very differently depending on the age of the subject?
Yeah, absolutely. Thank you for your question. So let me go back to the slides. Okay, so the model that previously fit very nicely the adult animals, it also fit our younger rat, only that it's not that the decrease in startle reflex doesn't appear here in the adult. We do see that, however, it's more prevalent in the younger animals. And so once again, with an added parameter of the adjustment of our formula or our model, is still be able to capture the PPI in the young animal fully, which right now we're trying to understand what it means in the context of neuropsychiatric diseases. Does that make sense?
Yeah. And you also do the experiment in humans.
Yes. Yes. However, we measure the PPI response in human a little bit differently. We use a tool that can capture the movement of the startle response underneath the eye instead of capturing the movement in the animal with the box.
All right. Thank you very much.
Thank you so much.
So we're running a bit behind. How about we take a 5-minute break? And then we will have our third session. And following our third and final session, we are hosting a reception. So please stick around. Thank you.
They're over, right? Okay, cool. Great. Welcome back, everybody, to the third and last session of the Intern Symposium, which has been great so far. Now it is my honor to introduce Michelle Taborga, who did her internship in the Krogan Lab under the mentorship of Ben Polacco. And she will be talking about, she was investigating peptides that have significant changes in mass spectrometry. And she'll be presenting about this. There you go.
Good afternoon. My name is Michelle, and I will be speaking about my bioinformatics summer internship here at the Krogan Lab, as was mentioned by Manon under Ben Polacco's guidance. So a little bit about myself. I'm a senior at Cal State University East Bay. I will be graduating this December with my degree in statistics with a data science concentration. What got me into the sciences was my love for math, and although I really like math, I didn't want to have it as like my major. So with more exploration, I found a love for statistics, and being that statistics is such a broad field, I was able to find this bioinformatics internship and learn what bioinformatics is, even though I had no biology background really before this. And bioinformatics is basically just at the heart of stats, computer science, and biology. The specific question I had in mind throughout my entire internship was, when are peptides significantly different or behaving significantly different? The data that I worked on was focused on APICS human cell data. And the way this data is collected is through an experimental proximity labeling method that identifies proteins near receptors at 5 different time points, time 0, 1 minute, 5 minutes, 10 minutes, and 30 minutes, once a drug is introduced. Then the protein and interactions are collected through this automated proximity proteomics, and using mass spec, you're able to identify these proteins and their peptides as well. Traditionally, the analysis of this data is done on the protein level, but I was interested to see if there was some findings that the peptide level analysis could give us that we were not seeing at the protein level analysis. The methods that I used was I computed linear model and ANOVA approach on each of thousands of peptides, proteins peptides for certain receptors. I did 2 approaches. I tried an interaction model, but that didn't work too well. Then I tried a 2-step process that basically involved subtracting the protein intensities from the peptide signal, and then performing that same linear model and ANOVA approach on protein detrended peptides this time. That did work a little better. I found some significant peptides, significant as in a p-value less than 0.005. And then once I found these peptides, I looked further into seeing what was causing them to be so different, maybe some possible phosphorylation sites on the protein/peptides. So this is kind of the idea behind the method. Basically, the original data shows non-significant peptides in gray, significant peptides in green, light blue, and red, and the protein is graphed there in dark blue. When the protein is detrended, then you would see this flat line when the protein is plotted, and if the peptides are still significant, you would still see them having their own trajectory, which they are. And then once we found those peptides having those own trajectories doing their own thing, then I would look into to see if maybe there are phosphorylation sites located there that may be causing these changes. Phosphorylation is a specific way that cell signaling works to turn on a protein or change its function, which is why this could be a possibility that this is affecting those peptides. So I performed a Fisher exact test on significant proteins and significant detrended peptides, and I found a positive correlation between the two. So peptide with changes are associated with protein interactors. And I saw this in both DOR receptor and QRFPR receptors. I was also interested, as I mentioned, to see the correlation between significant detrended peptides and possible phosphorylation sites. And I did see a positive correlation between the two when I looked at QRFPR. So there were more significant peptides found where there were possible phosphorylation sites. However, I did not see that same trend in the DOR receptor. I'm not sure why, but moving forward, when I looked into these receptors and actually plotted these peptides, first going over DOR, I saw that the peptides that did have significant changes, for example, in this protein CCDC88A, you can see the peptide in red is kind of doing its own thing. It shoots up like around time 3 and keeps going up. And there is actually known phosphorylation sites here, 1675 kinase CDK5 phosphorylates and 1690 kinase PKC phosphorylates. And there are also a couple other known phosphorylation sites. And although I didn't find that in my Fisher exact test, when I compared this, I did still see it in DOR. Moving on to this other protein that I found in the DOR receptor data, VPS33B. Same thing, you can see at time point around 3, the red peptide just kind of shoots up and continues on its own trajectory. This protein specifically plays a critical role in intracellular trafficking and vesicle-mediated transport within cells. And it is a DOR DOR activation triggers vesicle-mediated transport. So that might give us more of an insight. And as I mentioned, this one also has a phosphorylated site on Y312. The whole phosphorylation thing with DOR is just kind of a speculation since I don't actually see it when I did the Fisher exact test. So the fact that this is going up here at time 3 is just, there could be that it's increasing because there might be a phosphorylation site there. Moving on to QRFPR receptor, it's a G protein coupled receptor expressed in various tissues, including the brain, pituitary gland, and peripheral organs. I found this protein CTTN with 3 significant peptides that all seem to be having the same trajectory. And this protein is special in regulating the actin cytoskeleton, cell motility, and cellular adhesion. There were a lot of known phosphorylated sites on this protein, confirming my findings on specifically with this QRFPR receptor, that there is a relationship between significant peptides and possible phosphorylation sites. And as you can see here in this is the positions of all of the peptides in the protein plotted. You can see that all of the significant ones are all around the same position. This is another protein I found, PVR, and its role in cell adhesion, immune response, and virus entry has a very distinct trajectory compared to its protein. It's almost like perpendicular, and it has many known phosphorylation sites, again, confirming my findings for this receptor and possible phosphosites. Another example as well, this one is this GIGYF2 protein is involved in RNA metabolism and signal transduction pathways. Many phosphorylation sites on this protein, and also one ubiquitination site on K35. It could be possible that maybe the phosphorylation site is driving this ubiquitination site. I'm not sure. It's something we could look into. And then lastly, Cullen-3, this one was interesting to me because it did have known phosphorylation sites, and again, a ubiquitination site, which maybe it could be a driving force, the phosphosites for the ubiquitination sites. My conclusion is to apply this method to other receptors and expand possible applications of the method. I was only able to apply it to a couple of the receptors, so I would like to maybe apply it to all of them. And there is also maybe a possible phosphoproteomic experiment to validate the findings that I have found through this method. I want to thank everyone at the Krogan Lab, especially my mentor, Ben Polacco, and my computational team, who's really awesome, everyone in the Pumas and Gladstone as well. I've learned so much during my 2-something months here. So I can't imagine what I would learn being here longer. Questions? Thank you.
Thank you very much, Michelle. Any questions from the audience?
I was just wondering, have you thought about treating these? Yeah. Have you thought about treating the samples with a phosphatase to see if that kind of just removing phosphorylation groups recovers these peptides?
I have not, because I kind of learned everything about phosphorylation and everything during this internship, but that could be a good next step approach.
I was also wondering which type of tools you use to identify the phosphorylation sites on the peptides.
So I first, when I was crunching the numbers of data, I made a column when I found the peptide sequences. If there was an S, T, or Y in the sequence, I labeled it as a possible phosphosite. Once I plotted these peptides and I actually found like the significant ones, I went online and I researched to see if there were actual known phosphosites there. And that's how I kind of confirmed it. So first I kind of just guessed and then I confirmed it by researching.
More questions from the audience? Thank you, and we'll move on. Thank you very much again.
Thank you.
I guess while we're setting up here. Our next speaker will be Abhimanyu Ghosh. He did his internship in the Fraser Lab, sponsored by QBI and under the mentorship of Stephanie Vankovich. And he'll be speaking about parametric reduction of rotamer outliers with QFIT modeling. Take it away.
Okay. Hello, my name is Abhimanyu Ghosh. I am a rising 11th grader from Poolesville, Maryland, on a 6-week internship at Fraser Labs via the QBI program. Okay, so my presentation today is about the parametric reduction of rotamer outliers with QFIT modeling. Before we understand what QFIT and rotamer outliers are, we need to understand what protein dynamics is. Protein dynamics is a field in biology that is specialized in the flexibility of proteins and understanding the properties of it, including the cause. This field is beneficial as it helps helps with the understanding of interactions of proteins within a cell and with other proteins, which is also helpful in the medical field for drug development. Okay, so what we have here are conformations. Conformers are alternate variants of typical proteins which happen due to the flexibility of proteins. We can see on the right image of a side chain. The left image shows a protein side chain, the same protein side chain, while the right image shows the same side chain along with an alternate conformation of it. But it is still the same protein. Okay, so X-ray crystallography is a technique used by structural biologists to get a 3D picture of a crystal structure of a protein. This is done by blasting X-rays at millions of copies of a protein and looking at the diffraction patterns caused by the X-rays bouncing off the protein. However, because of the technique, the data is stacked on top of each other and must be analyzed manually for alternate conformations. So what is QFIT? QFIT is a computational software developed by Fraser Labs and is used in structural biology to help improve the modeling of a protein by computing multi-conformer side chains and backbones within them. Like I said before, this is something that's done manually, but now can be efficient and automatic. QFIT works by inputting a set of files containing atomic coordinates, which are the positions atoms are within a protein. These are taken from the protein data bank and are called PDB files. It also inputs a set of MTZ files, which contain X-ray crystallography models of the protein. QFIT then models the proteins in every possible configuration and scores the configurations based on how well they fit onto the electron density data. It displays the ones that fit best, which are considered to be alternate conformations. So if you go more in depth about it, QFIT first finds all the possible positions of the backbones within the protein using the protein data bank. It then scores the options and removes most of the options depending on how well they fit into the experimental data, with the more likely ones remaining as conformers. After finishing all the backbones, it moves onto the side chains and repeating the step. It moves with the side chains and does the same thing for the angles within the side chains, scoring the configurations and displaying them. So once once all possible backbones and side chains are analyzed, QFIT selects the best conformers and validates them based on how well they fit into the electron density data. Okay, so QFIT, as I said, displays the alternate conformers based on how well it fits into the electron density data, but sometimes there are exceptions or outliers called rotamer outliers. These are errors that happen in the computational model due to noise within the data and the flexibility of the proteins. So our overall goal is to decrease the number of rotamer outliers within the protein model, which we hypothesize we can do by altering two parameters, the rotamer neighborhood and the step size. So the image on the right is showing a chi angle, which is a sidechain angle that has every possible variant, which is shown in blue, along with a rotamer neighborhood, which is shown in orange. The rotamer neighborhood is the range which QFIT is searching, while the step size, which is shown in red, is the frequency which QFIT is searching within that range. So by varying the rotamer neighborhood and step size within the PDB models, we can reduce the number of alternate conformations. since a smaller neighborhood will allow for a greater area to be searched, and a smaller step size will allow for more frequent searches within the area. Okay, so based on the revisions, we have our conclusions, and if you can notice, is there a...
You can use the mouse. Oh yeah, you can try that actually. I think that should work.
Okay, if you look at the data, you can see that right here we have different outputs based on different amounts of step sizes and different amounts of road neighborhoods. You can notice that as the step size decreases, and same with the road neighborhood decreases, you can notice that there are a smaller amount of road outliers for all the structures. Whereas as you increase it, the amount of road outliers increases in both directions. So basically, more frequent step sizes and smaller road neighborhoods results in a smaller amount of road outliers. However, it is important to balance the amount of step sizes and road neighborhoods, because if they are too small, QFIT will take a lot longer to analyze them, which reduces their efficiency, and the proteins would become too rigid, which defeats the point of its flexibility, understanding its flexibility. Okay, I would like to thank QBI for sponsoring me, and Fraser Lab, especially my mentor Stephanie, and also Ashraya, for giving me this wonderful opportunity to learn about protein modeling and understanding what lab life is like. And I will always be grateful for this opportunity. Thank you. Any questions?
That's really interesting. Thanks. I was just wondering about when you're doing these calculations as to how the rotamers are positioned. Are you doing this on a single amino acid basis, or are you also taking into account how the other side chains around this amino acid are moving?
Oh, so what we did was basically we took several protein models, and we essentially analyzed all of the side chains, accounting for all the alternate conformations within the side chains.
But within these side chains individually, or kind of also alongside the neighboring side chains?
We did all the side chains.
Thanks.
In designing the algorithm, at some point, some conformations probably will disrupt the crystal structure itself and prevent the crystallization itself. So is there some limitation for like how much conformation can change where you still get good crystallization and a good crystal structure, or is that separate from the analysis?
I believe that the rotor neighborhood actually accounts for that, because like I said, like you have to try to balance between the rigidness of the proteins along with the amount of rotamer outliers that occur.
Are there other questions in the audience? I do have a question too. So I have two questions actually. One was that I noticed on the slides that it said new qubit for like it had new as the label here.
Yeah.
New QFIT model, and then I was wondering, is there an old one, and did you use it and compare it, or was the old one?
Actually, so yeah, I originally had old ones shortly before this, but the thing is, the old ones and the new ones aren't actually old and new because they're essentially comparing the same data. If you look at the actually, I wonder if I can show it on here. Probably not. If you look at the old one, it will just be identical to the new models.
So it was just a rerun, and
yeah,
called it that.
Yeah. So the new one, the new QFIT model was just a typo essentially.
Okay. And then I had another question, which is how many how many different protein structures you need to be able to use this? Because from what I understand is that you use available, like publicly available data or data that was collected in the lab previously, the PDB files, right? To input, and do you know like how many you need to be able to do some of that modeling there?
I am not sure.
Just one.
I believe you can do it with one. It's just that we essentially just took a lot of them just to make sure we had several examples.
Oh, okay, so but you did it separately and we don't, okay. You just need one crystal structure. Thank you. Great, thank you very much. And then, yeah, give it up to her again. And so we're moving on to our next speaker, which will be Nini Lee. Stephanie had her hands full this summer and had 2 interns because Nini was also, okay. Was also mentored by Stephanie in the Fraser Lab. However, she was sponsored through the AVID program. And students. And while they're setting up the talk, I guess I can first read her title. That's Impact of Distal Mutations to the Binding Site of Macrodomains and the Binding Affinity between them and drugs. And I wanted to take the time to say that the AVID program, which is the big center grant here at QBI, just like HARC had a program where postdoctoral scholars got a mentored scientist award. So Stephanie herself has won this award. And then this program contained the stipulation that they would also take an intern and mentor themselves. So it's like the mentors mentoring the mentors, or I don't know. There's a lot of mentoring going on. And Nini is the recipient of this mentoring from Stephanie. Okay. Yes.
Oh, wow. Thank you for the introduction. So I don't need to say any more about that. I mean, yeah. So I am Shovel College student and I will transfer into CSU is by this fall for biological science, concentration on cell and molecular biology. I would love to become the immunologist. However, during the time in the lab with the Dr. Stephanie, I figured that I enjoy pipetting, crystal harvesting, and especially craving for data analysis. I would love to explore more about computational structural biology. So the research in the lab is focused on fundamental principle of macromolecular structure and their dynamics. My specific project is to talk about macrodomain and the impact of the distal mutation to the binding site of macrodomain on the binding affinity between macrodomain and drugs. So why we care about macrodomain and what are the macrodomains? So macrodomains are the routine family that found in many different organisms from viruses to humans. They read and remove ADB ribose from the post-translational modification, which remove ADB ribose from the case signaling routine, which is the part of the, it's mean like they reverse the post-translational modification, which play an important role on oxidative stress, DNA repair, and immune response. So on the non-structural routine 3 of SARS-CoV-2 contain the macrodomains, which is MAC1, and on the SARS-CoV-1 occurred the threat to the public health in 2002, contain another macrodomain is highly related to SARS-CoV-2 macrodomain as well. So the scientists have worked a lot, and we already have the virus that can prevent the threat to the public health. But for our disease, we'll still need to find a drug that can target the macrodomains and inhibited the macrodomain by removing IDB ribose, so they can prevent them from reduce the innate immune response. However, so in conclusion, macrodomain will become the potential target for the antiviral drug developments. However, in our body, we also have the human macrodomains, and that are good guys, so we don't want to do something to them. So, so our goal is to design a drug that that target the viral macrodomain, but not human macrodomain. But not that's really easy. So, however, human macrodomain and viral macrodomains are really similar on the binding side, which binding to ADB ribose. But on the distal to the binding side, they are very different. And we would like to think about the different can drive into the different in binding affinity between them with drugs. So our hypothesis that changing a single amino acid with a distal to the binding site of the viral microelement can affect the binding affinity between them with drugs. We can see on the viral microelement Mark 1 on the location. My specific project is focused on the residue 144. I will make the which is distal to form the binding site highly related, highlighted in the red color, and I will change them to alanine, glycine, or phenylalanine or isoleucine. So like we can see, Joe of them is hydrophobic amino acid. So by compare volume to alanine or glycine, we can see alanine, glycine is less side chain, so they have more space to move in around. So maybe the entropy will decreasing, and maybe the entropy will be increasing because they can move in around, more flexibility, and the binding affinity will between them with drug will increase in. But for phenylalanine or isoleucine, we can see them more punky. It's really big with the ring and more side chain. So it's more make them more moving harder than volume. So it's reduce the entropy, like disorder, chaos to the system, and make them. We read it that the binding affinity when the drug will reducing compared to volume. So yeah, our method including those steps: first protein preparation, and then crystallization and data analysis. We first design the primers of the mutant and clone them in the vector backbone, and we got a plasmid DNA, and we're gonna build transform them to the D3 E. coli to express the routine. After we have the routine, we're gonna purify them and ready for crystallize them. When we have the structure of routine, we're ready for using the isotomal titration calorimeter to measure the protein and small to measure the binding affinity between the wild type and the drugs, and the mutation with the mutant with the drugs, to compare them to see if the binding affinity changing or not when we change in the single amino acid. So binding affinity was driven by the entropy and entropy. So using isotomal titration calorimetry, we can measure the binding affinity changing, entropy changing, and entropy as well. In summary, I will go to understanding genetic variation outside the binding site, influence the binding affinity of the macroelement to the drugs, and that can be used to develop the drug that inhibited SARS-CoV-2 macroelement, but not human macroelement, and also in different viruses in common as well, because COVID-19 is not the only one, not the first one, and might not be the last one, so be prepared. So we are, I'm successfully expressed the phenylalanine mutation, glycine, and isoleucine, ready for crystallization on the step of purify, still on purify them. Successfully have a crystal for alanine mutation, but I'm not the one who purify them. So for the future direction, we're gonna measure the crystal structure for each mutant and determine structure for the macroelement and compare them with the wild type, and using isotomal titration to measure the binding affinity of them, entropy and entropy, to see if the binding affinity changing or not. And we're gonna create more mutation in different single amino acids to support the idea that to support that hypothesis that changing the single amino acid can change the binding affinity eventually. And reference here, and I'm really grateful for BOOMAS programs and my doctors come here today, and my Fraser Lab, the people in the member in Fraser Lab, as well as my doctors, my mentor, Dr. Stephanie Arasa, coming in hierarchy over there for answer all my questions when I need help. And yeah, when I forgot everything, like one thing to remember on the lab is that one. Like I did not fail, just the experience fail.
Thank you. Any questions for Nini?
You talked a little bit about the differences between human and COVID macrodomain. I was just wondering how similar are SARS-CoV-1 and SARS-CoV-2 macrodomains? Do you know anything about this?
Oh, I think I was not that thinking about that, but it's really similar. Mentor.
Yeah, okay. Thanks.
Didn't know every answer. Other questions from the audience?
I might have just missed this, but I was curious if your interest in these like genetic variants was because there were variants in COVID macrodomains, or if it's like a pan-viral genetic variation.
I'm sorry, can you?
Your interest in these specific mutants, or is it because they're mutants in different variations of SARS-CoV, or because they're variations that are common across multiple different viruses that have macrodomains, or yeah.
Oh, like why we interested on residue 144?
Yes.
Oh, actually,
yeah.
Yeah, we are interested on the difference between the viral macroman and human macroman, and residue 144 is one of the differences.
Okay.
So.
Okay, thank you very much. You're going to be projecting from your computer, right? Okay, please welcome Angelica Marquez. She was doing an internship in the Krogan Lab, sponsored by the PUMAS program. She was co-mentored by Einar and Einar Kurtzetter and Michael McGregor lab, and she'll be talking about AD-associated ApoE4, investigating lysosomal dysfunction and calcium handling in neuronal cells. Take it away, Angelica.
Hi, everyone. My name is Angelica Marquez, and I'm a PUMAS intern for the Krogan Lab. And for the duration of my internship, I focus on Alzheimer's disease-associated ApoE4, investigating lysosomal dysfunction and calcium handling in neuronal cells. And so a little bit about me is that I've always been enthusiastic for the sciences. I really enjoy storytelling, whether that be from movies, TV shows, or books. Everything around us has a story, including our environment. And that's why I've always been interested in science, and most especially cells, because even though cells are the smallest unit of life, they make up all of the living organisms around us, and they can even tell us more about our environment. And so in high school, I enrolled in a lot of biology-related courses like biomedical sciences, human body systems, and biotech, and I enjoyed every second of it. So right now, I'm a biology major at Chabot College, and I hope to transfer in fall 2024. And so Alzheimer's disease is a neurodegenerative disorder, and I'll be focusing apolipoproteins known as the ApoE4 allele, ApoE allele, and ApoE2 is protective, ApoE3 is neutral, and ApoE4 is detrimental. So we hypothesize that ApoE4, a major AD risk factor, contributes to AD through altering the lysosomal calcium levels. And so something that ApoE4 does is that it does not degrade the amyloid plaques very well, which you can see as the yellow clumps, and it also does not degrade the neurofibrillary tangles as well. And so neuronal ApoE4 is associated with lysosomal dysfunction, which includes decreased acidity, respiration dysfunction, stress vulnerability, and impaired proteolysis. That's also related to the defective calcium handling. So the endoplasmic reticulum stores low calcium levels, and the mitochondria stores higher calcium levels. So in the diagram, you can see that the healthy lysosome would normally bind or fuse with the damaged organelles. However, the damaged lysosome does quite the opposite. So lysosomal calcium is vitally important because it allows the fusion between the damaged organelles and the lysosome, which maintains homeostasis within the cell. Normally, the lysosome will bind or fuse to the damaged organelle, which you can see that's inside, and that's all because of calcium channels or transporters, which is a calcium-dependent reaction. And so to start off with our experiment, we did gene editing. We took an ApoE gene variant, we took ApoE3, and we utilized CRISPR-Cas9 to insert a mutation of interest with the repair template, and we compared the two through a gel. And we did some prior testing first. We wanted to see which polymerase would be best to amplify our product. And one way to do this is to compare various polymerases through a range of temperatures. And the one with the most clean specific bands would accurately amplify our product. And once we had that, we utilized a restriction digest, HHAI, which only cuts ApoE4, and ApoE3 is left as its single band, which is, as you can see in the gel, it has that distinct clear band compared to ApoE4. A next experiment we did is calcium imaging, where we transfected Neuro2A cells with LAMP2 GCAM6S, and whenever that's in the presence of calcium, it'll glow, and we looked at that under a confocal microscope. And so this is a time-lapse video of Neuro2A cells that, oh, there we go. We inserted GPN, which releases the lysosomal calcium about right now, and then ionomycin releases the total amount of calcium within the cell, which you can see there. And then we can also watch it again. And if you notice that E3 has the brightest signal compared to the other two. And as a result, we saw that ApoE4 does not associate with the release of the lysosomal calcium, and E3 has the highest calcium signal, and E4 and wild type generally have similar levels. We also inputted our data onto a graph, and you can see that the blue line, which is the ApoE3, tends to have the highest amount of calcium through time. And again, GPN releases only the lysosomal calcium, and the ionomycin releases the total amount of calcium. And so for our next method, we did lysosomal density. We did an immunofluorescence, and we utilized DAPI for nuclear identification, and LAMP1, which are antibodies that attach to the lysosomes and it'll glow. And we also looked at that under a confocal microscope. And as a result, we took a sample area for each of the alleles, and we counted each and every single green dot that you can see, because that is a single lysosome. And then we input it all into a graph, and you can see that ApoE4 has a significant amount of lysosomes compared to both E3 and wild type. But we only wanted to focus on the single lysosome calcium results. And to do that, we took the area under the curve for GPN and divided it by the area under the curve of ionomycin, which is from the previous calcium graph, to only normalize the calcium sensor expression. And with that data, we divided it with the previous graph I've shown earlier with the lysosomal density, and that will only account for calcium per lysosome. As you can see that each of the alleles have a similar amount of levels. And so for future directions to further this experiment, we could develop pure ApoE4 shy cells to find more cell-related risk to AD, as well as incorporate novel ApoE4 screening protocols similar to the RFL-PSA, and identify calcium-independent mechanisms of lysosomal dysfunction, which during this experiment, we did calcium-dependent mechanisms. And we could utilize also additional studies with lysosomal calcium, especially with mature neurons such as Neuro2A cells or even other cell types. And in summary, I did an RFL-PSA, including PCR and gel electrophoresis, while also utilizing CRISPR-Cas9 to support successful gene editing of ApoE4 cells. And we found that ApoE4 expressing neuronal cells does not cause an accumulation of lysosomal calcium, but it does have an increased amount of lysosomal density. And in conclusion, our findings suggest that neuronal ApoE4 cells accumulate lysosomes with unchanged lysosomal calcium levels. And so I would like to acknowledge the UCSF Krogan Lab, Gladstone Institutes, the PUMAS program, and special thanks to my mentors, Einar and Michael. Thank you for your time, everyone.
Great job. Thank you very much. Does anybody have a question?
So with the finding of the increase in the number of lysosomes without themselves changing, is the thought that that's responding to something, or that's itself kind of the causative problem? Like, is it they're increasing because they're bad in other ways, or something else is happening? Do you have a sense of why they're increasing?
I'm not too sure of why they're increasing. I think that it's really interesting that because E3 has more calcium, you would think that it would have more lysosomes, similar for E4, which has the most lysosomes detected, but it does not have an increased calcium levels. I'm not too sure of why that's the case.
So I guess the higher lysosome that maintain similar lysosome.
Hey, great talk. In addition to the difference in density, do you see any other differences like morphology or anything? Can you tell if the lysosomes look different in the two different cell types?
During the experiment, we didn't really focus on like any like size differences or any other factors but that's really something exciting to look onto to further on this experiment.
Great, thanks.
Questions? I would have a question also, again, about, I think, software that you use. I was wondering, most probably you didn't count the lysosomes by hand.
No.
Do you want to comment on this, how this is done?
Yeah, I actually thought I asked my mentor like how would we be counting each of every lysosomes. I thought I would have to count it individually one by one and like draw a box, but no, that wasn't really the case. Instead, we used the Fiji program, and all I did was just sort of trace around each cell, and then did some Excel and like some coding, and it just like accounted for it all onto like an Excel sheet really easily.
Yeah, that's what I thought, but I thought it would be nice to share. Okay, thank you very much. I should stand closer here. We're moving on to our next speaker. Be coming up. Next speaker is going to be V Trong, and she. Oh, I'm sorry, that was still from Angelica, so she stayed within her time. V is also a PUMA student, and she actually is not associated with QBI at all. She independently contacted us and wanted to present here, and I think I wanted to note, say that because I think it's great. We're very happy to have you. She did her internship in the Theodoros lab and was mentored by Bumjun Kim. And she's going to be speaking about induced pluripotent stem cells to, oh, I forgot the first part, locating specific gene expression during induced pluripotent stem cells to cardiomyocyte differentiation. And now we have to figure out how to get it over there. Let's see. We did this before. Gina, do you need to press something, or will it do it automatically, or you're not plugged in? Okay, that's the problem. Okay. Sorry. Over here. Yeah. Okay. Okay, that looks better. Okay, take it away.
Okay.
Hi, everyone. My name is V Trong. I am a third year at Los Mananos College, and I'm hoping to transfer in 2024. This summer, I had the greatest opportunity to be part of the Puma's internship program. During the course of my time here, I've learned a lot and gained a lot of information in such a little amount of time of about 9 weeks. Through although it felt like time has passed by really quick, the information that I've gained is worth about maybe like a year or so of practice and education. But luckily, I was under the mentorship of Dr. Bum Joon Kim. He has taught me from the simplest things like opening a vial with one hand. I know it doesn't look very cool, but it took me about a week to get that down. And also pipetting microliters of water from one dish to another, and all the way to growing my very own cells in vitro. So here is what I have dedicated my entire summer to. So I have worked on locating specific gene expression during induced pluripotent stem cells to cardiomyocyte differentiation. So in 2007, our very own Gladstone employee, Shinya Yamanaka, discovered induced pluripotent stem cells technology, which allowed scientists to convert somatic cells like blood cells or skin cells or anything else into iPSC by just adding 4 factors called the Yamanaka factors. That is OCT4, SOX2, c-MICE, and KLF4. In the past, people have gained PSC, pluripotent stem cells, from embryos. And as you can see, that rose a lot of controversies. So now that Mr. Yamanaka has figured out a different way to acquire PSC, we can have other options other than acquiring it from embryos. So why iPSC are important and what we can use it for is for pharmaceutical drug discovery and also safety toxicology. So the methods that I went about differentiating my iPSC into my cell type that I grown, which is cardiomyocyte. Cardiomyocytes are responsible for the pulsing of your heart. So I used a STEM diff kit provided, and I followed the exact protocol that they provided for me. So it took about 14 days, very quick, to turn an iPSC into a cardiomyocyte. So first, after the 14 days, I've taken I extracted RNA from each of my harvested cells, converted it into cDNA, and then put it through a qPCR process. The reason why I put it through qPCR is because it's important to pinpoint the gene expression to tell us which stage of differentiation our cells are in. For example, if it is expressing NANOG, then it is in the stage of iPSC. If it is expressing DKK1, it is a mesoderm, and so on and so forth, to CTNT, which is cardiomyocyte. So here are the results that I've acquired during my differentiation process. On day -3, here are some microscopic photos that I've taken directly from my cells. Day -3, we see it is an iPSC stage. And then on day 0, which is when the differentiation process started, this is when it reached 95% confluency, which means that it has taken up 95% of the cell plate itself. On day 2, it has gained a more 3D layer rather than its 2D layer on the cell plate. So on and so forth until day 14, I started to notice that the cells have began to pulse. You can see here, this is just the regular cell under a microscope. It's starting to show the morphology of pulsing.
And then here is the second picture. It's the same photo, but just under a UV light. So the cell line that I've used contained GFP. GFP is green fluorescent protein. Green fluorescent protein emits a green fluorescent from the protein myo7, the myosin light chain. This is the myosin light chain protein is only present in cardiac muscles. So we don't know exactly, we cannot say that it is exactly what it is just based on its morphology. So with my results from qPCR, here is why I can be sure, or a scientist can be sure that it is what it is. I have color coordinated it. Green is green in the beginning, blue is for mesoderm. So in the beginning processes, what I would expect is for iPSC to show up more greater compared to the other cell types. And then so on to the end here where cardiomyocyte should be expressing TNNT, which should show up here. Clearly, that is not what is shown. So after running through qPCR of a day close to my poster session, which is the end of my internship, I realized, oh no, I do not have time to restart this entire process again. So I felt really like defeated in a way, especially because I wanted my results, you know, like I've got the morphology correct. However, the data did not come out the way I expected. From feeling defeated, I've talked to my mentor, my internship friends, and my lab about it. I was like, oh no, like, what do I do? I don't have accurate results. This is so embarrassing. But they've told me, they reminded me that this is all part of science. This is my first experience in a lab itself. So I just have to trust in the process. No experiment is going to go perfectly the first time. So that gave me a lot of confidence. And I just want to continue that on to honorably mention the people throughout this process that gave me the confidence in my scientific journey these 2 months. So I would love to thank my mentor, Dr. Bumjun Kim, for reminding me the values of what I'm doing. And then I would love to thank my Pumas program for giving me the opportunity in my first lab experience ever. And then I would love to thank my Theodorus lab for inviting me into such a friendly environment and creating a safe space for me to make mistakes and grow in the way that I grew throughout these time that I have here. So thank you so much for being here and listening to me.
Thank you very much, Bea. Does anybody have any questions for her? I can start off with one. Yeah. I was wondering, so for it seemed like you most probably differentiated into cardiomyocytes, right? Because they were beating and not most cells don't beat.
Yes.
And so I was wondering if you had any controls in your qPCR reactions and what that might have told you about where things might have gone wrong.
Well, the qPCR process was done a day before the poster session, and because of the time limitation, we can only run one qPCR. And unfortunately, yes, it didn't turn out correct. However, during the poster session, I had a lot of commentary on why that could be that it had gone wrong. The first thing that somebody told me was during the plating process, bubbles are not good at all because it deflects the probe that would scan the mixture itself. And it's okay. And unfortunately, yes, I did have bubbles now that I think about it. So that's one thing. Another thing could be anything really, like contamination or anything that could happen. Yeah.
the timer went off. I do want to, if anybody has an urgent question for me right now, feel free to ask it. I did want to mention we have the reception coming up. I wanted to, before you guys all leave, ask all the presenters to please meet somewhere, I guess, by Gina up there, because we would like to take a picture of everybody together so that this is all nicely documented. I wanted to take the time to thank all the presenters again. I think this was amazing. It was very interesting to learn about all of your experiences, and I think everybody did a great job, and it was very clear. Yeah, thank you. I think we didn't get the second applause. Yeah. No, I'm sorry. I lost my train of thought. It was great. Everybody had a very clear presentation. I learned a lot, and I hope that everyone in the audience also enjoyed it. I hope that people on Zoom also got something out of it. I did not monitor this. I think Gina was on this. We forgot to ask for questions there. Next time we'll do that a little better. There's always ways to improve. I mean, maybe that's what everybody learns during an internship also. There's always like a little room for improvement to the top. Yeah, thank you very much again. And thanks also everybody for coming and supporting the presenters.